Welcome to Prospective Doctors MCAT Podcast, brought to you by Med School Coach. Each week, Sam breaks down the highest yield MCAT topics so you can score as high as possible on test day.
Hello and welcome to Prospective Doctors MCAT Basics with your host, Sam Smith. The goal of this podcast is to cover the highest yield topics on the MCAT and provide you with some sort of insight into the questions that the MCAT really likes to ask. This podcast is going to cover membranes. So in the first segment, I'm going to go over a bunch of different topics regarding membranes. I'm going to talk about what a membrane is, its structure, its function, the various types of membranes in a cell, and then lastly, I'll talk about cell-to-cell junctions, since in some ways they involve the plasma membrane. After that intro segment, I'll go into membrane transport, then membrane proteins, membrane dynamics, and last I'll talk about membrane potential. And this is going to be a lot of information coming in pretty hot, so obviously feel free to pause it, rewind, listen to it multiple times, whatever you need to do. And this material will show up on 2 of the 4 MCAT sections. That's going to be biochem, and the chem physics sections. That said, I hope you find this podcast helpful, and let's get into it. As I said in this intro part of the podcast, I'm going to go over a lot of different things, starting with the broad definition of what a biological membrane is. Biological membranes form semi or selectively permeable barriers between different compartments in a cell. And there's a multitude of different membranes in the cell, right? There is a membrane that separates the inside of the cell from the outside of the cell. It's called the plasma membrane. There are 2 membranes in a mitochondria. There's an inner mitochondrial membrane, and there's an outer mitochondrial membrane. The endoplasmic reticulum has a membrane. And so there's just a lot of examples of membranes inside the cell. So overall, biological membranes are these selectively permeable barriers that compartmentalize the cell, and in many ways allow it to have a multitude of different organelles. So let's talk about the composition of a biological membrane. So it's made up of a phospholipid bilayer. And a phospholipid is this molecule that contains 2 fatty acid tails or chains that are linked to glycerol, and then the glycerol is linked to a phosphate group. And you're probably used to looking at these like molecular representations of them that look kind of like tadpoles. And the heads of these tadpoles are the phosphate groups, and these are all hydrophilic. And so they all line up together, and those are what faces the aqueous solution, whereas the tails are very hydrophobic. Those all line up together because they want to get away from water. And so it forms a bilayer because the tails of these adjacent molecules line up. So you'll have tadpoles that are essentially facing opposite directions. The tails are touching each other, and that's what creates that phospholipid bilayer. There are also some other components to biological membranes that you should know. The first being sphingolipids. So sphingolipids are lipids that contain a sphingosine backbone. And it looks actually fairly similar to a phospholipid, but you can recognize a sphingosine backbone by looking for a nitrogen in the head group. So you look up, you see nitrogen, and it's usually in the form of an amine group or an amide group, and you say, okay, that's a sphingolipid. It's got the long fatty acid tail, and then in the head, it's got the nitrogen. So that's how I recognize it. And another really important component of biological membranes is cholesterol. Cholesterol is this hydrophobic molecule, contains lots of carbons and hydrogens, and it's got these 4 rings that make it pretty recognizable. And cholesterol is really important for maintaining the fluidity of the membrane. Biological membranes can also have carbohydrates, so glycolipids are lipids with a carbohydrate attached by a glycosidic bond, which is just a covalent bond. And in terms of what this looks like in the membrane, the lipid part of the glycolipid is what actually sits inside of the membrane and kind of anchors the glycolipid into the membrane. And then the carbohydrate part of the glycolipid is kind of sticking out of the membrane. Most of these sugars are going to be found on the outside of the membrane. So for example, in the plasma membrane, carbohydrates are mainly on the exterior of the cell and can be used for many different purposes, including facilitating cell recognition. For instance, if you're a person with O-type blood, you're going to have red blood cells that have a very specific glycolipid. And this glycolipid is going to have a sequence of carbohydrates being glucose, galactose, N-acetylglucosamine, N-acetylgalactosamine, and then fucose. And then membranes also contain membrane proteins, and I'll talk about those in greater detail later in the podcast. So all in all, there's a bunch of different components that make up a biological membrane, but it's all rooted in this phospholipid bilayer. All right, so the next thing I want to talk about here is how membranes form. So lipid bilayer formation occurs spontaneously. The process of lipid bilayer formation has a change in Gibbs free energy of about negative 25 kilojoules per mole. So it's pretty freaking spontaneous. And as I said earlier, essentially what happens is that the heads of these phospholipids are hydrophilic and the tails are hydrophobic. So the heads all line up together because they prefer this aqueous solution, whereas the tails all kind of line up together because they prefer not to be near aqueous solution. They prefer each other. And this ends up forming the lipid bilayer. And biological membranes are formed by adding to pre-existing membranes. So in eukaryotic cells, membrane synthesis occurs in the endoplasmic reticulum, and essentially new membrane lipids are created in the smooth ER, then they're transported through the secretory pathway and are distributed to the plasma membrane and the other membranes throughout the cell. And I will also say that individual phospholipids can actually flip back and forth from being on the exterior of the membrane to the interior, and this is catalyzed by two different enzymes with very cool names. Flipases are transmembrane enzymes that catalyze the transfer of phospholipids from inside the cell to outside the cell. So just imagine the phospholipid with its head inside the cell, all of a sudden flipping to the outside of the cell. In contrast, flopases are enzymes that catalyze the transfer of phospholipids in the other direction, so from outside to inside. So in this case, you can imagine a phospholipid with its phosphate head pointing outside the cell, and all of a sudden it flips over to the inside of the cell. And these are fairly easy to remember. I just remember flipase has an I, so that's flipping phospholipids from the inside to the outside. Flopases, which has an O in it, are moving phospholipids from the outside to the inside. So that's a little bit on how these biological membranes are formed and altered. Next, I want to talk about some of the biological membranes that you will see in a cell. First and foremost, and also the most famous membrane of the eukaryotic cell, is the plasma membrane. And so there's been an interesting model that's been proposed to kind of describe the dynamic structure of the plasma membrane and more of the dynamic nature of the plasma membrane, and that is the fluid mosaic model. And the fluid mosaic model essentially says that if you were to take a little section of the plasma membrane, it would look very heterogeneous. It wouldn't just be these phospholipids. There'd be lipids, there'd be cholesterol, there'd be proteins, and they would all be kind of intermingled to make up the plasma membrane. And that's where they get this term mosaic. As Wikipedia says, a mosaic is basically a picture or a decoration made up of small, usually colored pieces of stone or glass. And so because of all these various components of the membrane, it kind of looks like a mosaic. And then they added this fluid part to the fluid mosaic model because if you zoomed into this plasma membrane, you'd see that it is not static, right? It's a very dynamic membrane. There's all these different components and they're all kind of moving around, right? You'd see carbohydrates whizzing by and proteins sliding past each other and all these lipids moving past each other all at once. And so it's not this static thing where you have like a protein just sitting there in the membrane, same exact location all the time, right? It's constantly moving. And if you were to keep looking at this plasma membrane, eventually you would notice what are called lipid rafts. And these are small bits of the plasma membrane that are enriched in cholesterol and sphingolipids, and sometimes they have proteins anchored to them. And quite literally, they look like rafts floating on a lake, where the lake in this example is the plasma membrane. And one thing that I thought was really interesting, I was reading this Nature review paper about lipid rafts, and it says that, quote, "These domains still elude direct in vivo visualization." In vivo meaning in animal compared to in vitro, which is in test tube. What I'm taking this to mean is that they can't actually be visualized on the cell. So you can take these cells out of the body and you can purify these lipid rafts from the membranes. But if you were to take these cells and, point a really strong microscope, maybe like a electron microscope or something, that you can't actually see these lipid rafts. Which is kind of interesting, right? Because we have pretty good technology to image cells. You'd think we'd be able to see these. And by the way, if I'm getting that wrong and you're like a lipid raft expert, first of all, bless your soul. Second of all, I'm sorry, not an expert, but that's just kind of how I'm taking that. And anyways, so I'm not really sure what to make of the fact that they've never been able to actually observe these on cells. It just makes me think, well, Is this something that's being created in the process of breaking down these membranes and purifying the different proteins and things from the membrane? Is that just an artifact of that? And of course, I'm sure that there's good evidence to say that that's not the case, but just still very interesting that scientists have never been able to see these lipid rafts in vivo. And these lipid rafts, if they exist, are suspected to play a role in signal transduction. Obviously, proteins can be anchored to these lipid rafts, and those proteins can be receptors, so that's how they might play a role in signal transduction. They also might play a role in secretory endocytic pathways, things involving membrane trafficking. So that's a plasma membrane. Of course, there are a lot of other organelles in a eukaryotic cell that have membranes. I'm sure you've heard the term membrane-bound organelles, which are what set eukaryotic cells apart from bacteria cells. And these organelles include the mitochondria, the nucleus, the endoplasmic reticulum, the Golgi, vacuoles, lysosome, chloroplast, and that's just to name a few. The ones that I want to give a little bit more detail on are the ones that I think are most important for the MCAT, and that's going to be the mitochondrial membrane and the nuclear membrane, or the nuclear envelope. Let's start first with the mitochondrial membranes. So that's an important note. The mitochondria has two membranes. It has an inner membrane and an outer membrane. So the outer membrane separates the intermembrane space from the cytoplasm. So that's that first membrane. Like if I was to take a mitochondria and stab it with a knife, the very first membrane I'd go through is this outer membrane. And again, that separates intermembrane space from the cytoplasm. The inner membrane separates the intermembrane space from the mitochondrial matrix. And this inner membrane is where the electron transport chain is embedded. And this is just illustrated by the point that if you look at the protein to lipid ratio of the inner mitochondrial membrane, you'll see it's about 80-20. Whereas if you look at the protein lipid ratio of the outer membrane, you'll see it's about 50-50. And that big difference between those two membranes is due to the fact that the electron transport chain is embedded into the inner mitochondrial membrane. And I will mention too that the outer mitochondrial membrane is a lot more porous. So it's got these porins, or these channels that allow ions and other small molecules to pass through with ease. And that's in contrast to the inner membrane, which is a lot more selective. And there is good reason that this inner mitochondrial membrane is so selective. So throughout the oxidative phosphorylation process, you're pumping hydrogen from the mitochondrial matrix into the intermembrane space. And what you're doing there is you're building up this electrochemical gradient of hydrogen ions. If this membrane was to be very permeable, well then you would just dissipate that concentration gradient because these hydrogen ions would just flow through, and you don't want that, right? What you want to happen is you want these hydrogen ions to flow through the ATP synthase, and you want to use that electrochemical gradient that you've created through this oxidative phosphorylation process in order to drive the production of ATP. So that is the two mitochondrial membranes. The next thing I want to talk about here is the nuclear envelope. So the nuclear envelope is the membrane that separates the nucleus from the cytoplasm. And it actually contains two separate membranes, and they're connected. If you go look up a picture, you'll see that it's kind of a fold over of the membrane. And so you have an inner membrane and an outer membrane. And between those, you have the perinuclear space. And studded throughout this membrane, there's a bunch of these little protein structures called nucleoporins, and those are very important. They are small channels that allow for the bidirectional exchange of macromolecules between the nucleus and the cytoplasm. And this is going to include things like proteins, RNA, and ribonucleoprotein complexes. And this nuclear envelope plays a pretty important role in regulating translation. So again, translation is the conversion of mRNA to protein by ribosomes. So there's ribosomes that are located in the cytoplasm and also bound to the rough ER. And there's not ribosomes inside the nucleus. So in short, these nucleoporins can regulate the rate at which mRNAs make it out of the nucleus and into the cytoplasm where they can then be translated. Hence, they control the rate at which different genes get translated, and therefore they can control gene expression. And it has also been shown that nucleoporins can actually associate with the promoters of some genes. And it's also been shown that nucleoporins can associate with the promoters of some genes and actually drive transcription of that gene, or act as a transcriptional activator. So there's two different ways really where it can regulate gene expression, either at the transcriptional level or the translational. All right, so that is the nuclear envelope. I just want you to keep in mind that there are membranes, biological membranes, all over the cell, right? I just gave you a few, and these have different functions depending upon where they are in the cell, depending upon the proteins that are embedded in the membrane, like the nucleoporins or the electron transport chain proteins. And, they're not these static structures, right? They've got parts that are moving all around, proteins, carbohydrates, different types of lipids, and it's a very dynamic, moving, fluid structure. The next thing that I want to talk about here are cell-to-cell junctions. And I will mention this is different than cell-to-cell communication, because the first thing that comes to my mind when I think about cell-to-cell junctions, for whatever reason, my mind goes to the communication part of like juxtacrine, autocrine, paracrine. This is different. And you also might be thinking, this is a membranes podcast. Why are you talking about cell-to-cell junctions? And I'm talking about these because it involves membranes. Duh. So there's 3 types of cell-to-cell junctions, and this is based upon the function of the junction. And these 3 types are anchoring junctions, communicating junctions, and occluding junctions. So the first type, anchoring junctions, are junctions that mechanically attach cells together via their extracellular matrix. So these are things like adherins, desmosomes, and hemidesmosomes. The second type of cell-to-cell junction are communicating junctions. So these let molecules pass freely between cells. So this is things like gap junctions, which allows the cells in the heart to contract in a synchronized manner. And then lastly, you have occluding junctions. And these are junctions that seal cells together in an epithelial layer in order to prevent molecules from leaking through. So an example there is something like a tight junction. So to quickly summarize, anchoring junctions attach cells together. Communicating junctions allow molecules to pass between cells for communication purposes. And then occluding junctions seal cells together to create a layer of cells that doesn't allow the passage of certain molecules through. All right, so in the next part of this podcast, I want to talk about transport through a membrane. So in my opinion, there's 2 things to know about transport through a membrane. The first being what types of molecules can pass directly through the plasma membrane and why. And then number 2 being how are molecules transported through the membrane that cannot diffuse straight through. So some molecules can pass fairly easily through the plasma membrane. Others have a fairly difficult time doing so. And so what determines this? There's 2 different factors. The first being size and the second being polarity. And I discussed this in detail in the biomolecule structure podcast. So if you want to hear more about this, go listen to that. But again, the size and polarity of a molecule are what determines whether or not it can diffuse straight through the plasma membrane. So only relatively small, non-polar molecules can move through the lipid bilayer. That's going to be things like oxygen, carbon dioxide, and lipids. Now that means polar molecules and very large molecules have trouble passing through the lipid bilayer without some other kind of assistance. So things like amino acids, glucose, and electrolytes have trouble diffusing straight through the plasma membrane. So let me quickly summarize that for you. Small, non-polar molecules, large, polar molecules. Now with that said, there are ways in which we can transport these polar or large molecules across the membrane that won't make it through just on their own. And these modes of transport are broken up into 2 different categories: active transport and passive transport. Active transport uses ATP to move molecules against their concentration gradient. Passive transport, on the other hand, is a mode of transport that does not use any energy and actually allows molecules to flow along their concentration gradients. And there are 2 types of active transport, the first being primary and the second being secondary. So in primary active transport, you're using ATP directly to move molecules against their concentration gradient. So this would be something like a sodium-potassium pump, right? You're moving 3 sodium out of the cell, 2 potassium into the cell, and you're doing this through the hydrolysis of ATP. Secondary active transport, on the other hand, uses another molecule's concentration gradient that has been built up by ATP in order to transport a molecule against its concentration gradient. So take, for example, the sodium-potassium pump that we just talked about, right? You're taking 3 sodiums, moving them out of the cell, 2 potassiums, moving them into the cell. And what this does is it creates a concentration gradient of sodium and potassium. Now, the sodium glucose cotransporter 2, or SGL2, is a transporter that's found in the enterocytes of the small intestine and is involved in the uptake of glucose from the food that we eat. And what this SGLT2 protein does is it uses this sodium gradient that's been built up by the ATPase pump, the sodium potassium ATPase pump that we just talked about, in order to move glucose against its concentration gradient. And so what this SGLT2 transporter is doing is it's taking a glucose and a sodium, which are each in the lumen of the gut, and it's bringing those in together into the enterocyte. And this is moving glucose against its concentration gradient and sodium with its concentration gradient. So you can see that the sodium concentration gradient is essentially being used to drive the movement of glucose against its concentration gradient. And I should mention that there are two different types of secondary active transporters. There are antiporters and symporters. Antiporters, which are also called exchangers or countertransporters, move a molecule against its gradient at the same time they displace one or more ions along its gradient, and in this case, the molecules move in opposite directions. So this would be like glucose goes into the cell, sodium goes out of the cell. But as I said, that's not the case. In the case of the SGLT2 protein, this is a symporter where molecules move in the same direction. So this would be where you're displacing one molecule along its concentration gradient, the other against its concentration gradient, and these molecules are moving in the same direction, like both into the cell, like in the case of the SGLT2 transporter. So that's active transport. Again, that requires energy. There's also passive transport, and there's a few different types of passive transport. So there's simple diffusion, facilitated diffusion, and then osmosis. Starting with simple diffusion, this is just the spontaneous diffusion of a compound across the plasma membrane without the use of any transporter proteins. So this is what I was talking about earlier when I was saying these really small non-polar molecules are able to just diffuse across the membrane. This is going to be things like oxygen, lipids, carbon dioxide. Facilitated diffusion is the spontaneous diffusion of a compound across the plasma membrane with the use of a transporter protein. So basically, this is just diffusion of a compound that needs a little bit of help getting across the plasma membrane. And I will mention too that this is the movement of a molecule down its concentration gradient. Obviously, to move against concentration gradient, you need energy, and that's active transport. But for passive transport and facilitated diffusion, this is down a molecule's concentration gradient. So let's take another transporter that transports glucose, this time the GLUT1 transporter. So this transports glucose across the plasma membrane into cells. So from the plasma into cells where it can be converted into ATP. And since the cell is constantly converting this glucose into ATP, the concentration of glucose in the cell is relatively low. So therefore, moving from the plasma into cells, that is down its concentration gradient. And this is essentially facilitated by this GLUT1 transporter. The last type of passive transport I'm going to talk about is osmosis. And this is the movement of water across a membrane from areas of lower concentration to higher concentration. In other words, this is when water moves across a membrane in order to even out the concentration of some molecule on each side of the membrane. And I'm not sure osmosis is all that important for an actual real biological system, especially compared to these other modalities, but I do know that I've seen a good example of this in the lab. If you've worked in the lab and specifically worked in cell culture, you know that you never add pure water to cells when you're culturing them or when you're washing them. Well, why is that? Well, you would know that if you added pure water, it doesn't have any solutes in it. And so when you add that to a cell, what's going to happen is that through osmosis, the concentration outside and inside of the cell are going to try to even out. What's going to happen, you're going to get a bunch of water that enters the cell, and it's actually going to cause your cells to expand and burst, and then you're not going to have any cells, and that's going to be because of osmosis. So that's a little bit on the different types of transport. I want to talk about next what these membrane transporters look like. So membrane transporters in general are multi-pass transmembrane proteins, meaning that they span the lipid bilayer multiple times. And there are two main classes of membrane transporters. There are carriers and there are channels. And they're essentially what they both sound like. So carriers, quote, bind the specific solute to be transported and undergo a series of conformational changes to transfer the bound solute across the membrane, end quote. And that is from the textbook Molecular Cell Biology. And so these carrier membrane transporters, essentially, you can imagine these like a non-continuous transport, right? Because it has to actually bind the solute and then have a conformational change and then bring that solute to the other side inside the cell. And so an example of a carrier membrane transporter would be the GLUT1 transporter, right? It's got to bind glucose, have a conformational change, and then spit glucose out inside the cell. The other type of membrane transporter is a channel, and from the same textbook, it says that channel membrane transporters, quote, form aqueous pores that extend across the lipid bilayer. When these pores are open, they allow specific solutes, usually inorganic ions of appropriate size and charge, to pass through them and thereby cross the membrane. And so I like to think about these as more of like a continuous transport, right? When these pores are open, the ions can flow through them continuously. It's not like, you can only do one at a time. This is just continuous transport. And something like an aquaporin would be an example of a channel membrane transporter. So to quickly summarize all that, only small nonpolar molecules can pass through the lipid bilayer by simple diffusion. Transporting molecules against their concentration gradient requires energy in the form of ATP or some other concentration gradient that's been created by ATP. And then membrane proteins either fall into the category of being carriers or channels.
Thanks for checking out the Prospective Doctor MCAT Basics podcast. Sam's doing a killer job taking you through the most important MCAT topics, but what if you need a little extra help? How does a 5, 10, or even 15-point increase in your score sound? Imagine how your chances at admission could increase. Med School Coach's MCAT tutoring can get you there. With the most rigorous selection process of any tutoring company, we see amazing results. We deconstruct each student, find a plan that is going to work, and help execute it. That's why our students add an average 12 points to their score. Completely physician-run and operated, and focusing on nothing but medicine. It's no wonder over 10,000 past students have trusted Med School Coach to get them through the MCAT and into medical school. Check out medschoolcoach.com today and mention code PODCAST for 5% off.
All right, the next thing I want to talk about in this podcast are membrane proteins. And membrane proteins can be broken down into 3 categories based upon their location in the membrane. So there can be peripheral membrane proteins, integral membrane proteins, and lipid-anchored membrane proteins. Integral membrane proteins have one or more segments embedded in the plasma membrane. This would be like the membrane transporters that I just discussed. And essentially, you would need to destroy the membrane in order to remove these proteins. And the transmembrane domains of these integral proteins, or the domains that actually go into the membrane, are typically either beta barrels or alpha helices. In addition to that, the membrane spanning portion of this protein, again, which I called the transmembrane domain, has side chains that are hydrophobic residues. And that makes sense, right? Because inside the membrane, there are all these fatty acid tails that are nonpolar. So you're going to want to have these hydrophobic residues in order for there to be a favorable interaction for that transmembrane domain to kind of just sit inside the membrane there. Peripheral membrane proteins do not interact with the lipid core of the plasma membrane and are usually bound indirectly to the membrane by interactions with integral membrane proteins or with these phosphate polar head groups of the phospholipids. I think the really big takeaway for peripheral membrane proteins is that they're reversibly attached to one side of the plasma membrane and that they don't enter the membrane. And this is all according to the Molecular Cell Biology 4th edition textbook, because you can read some different things on like Wikipedia, for instance. And there's a few interactions that keep these proteins loosely attached to the plasma membrane. First of all, there are van der Waals interactions, so you can have hydrophobic interactions. And then you can have electrostatic interactions where you have like one charged integral protein, or maybe that has like a charged residue on the outside. And then the peripheral membrane protein also is charged, and so then you have an interaction there that kind of loosely ties it to the plasma membrane and makes it a peripheral membrane protein. Now, the third category of membrane proteins are lipid-anchored proteins. And these are proteins that are covalently bound to lipids that are then embedded in the plasma membrane. And some sources I looked at categorized these as peripheral. Some of these sources I looked at categorized these as integral membrane proteins. So I'm kind of making this its own category. I guess depending upon the source you look at, it'll either tell you it's integral or peripheral. But I think the big takeaway is that a lipid-anchored protein is just a protein attached to a lipid covalently, where the lipid is then inserted into the plasma membrane. And overall, there are 3 different types of lipid-anchored proteins. There are prenylated proteins, which are just proteins attached to isoprenoid groups, which are these large nonpolar groups. There are proteins linked to these long fatty acids, which are fatty acylated proteins. And then you have GPI proteins, or glycosyl-phosphatidyl-lynosyl linked proteins. In terms of the functions of these 3 different types of membrane proteins, for the most part, integral membrane proteins tend to either be cell surface receptors, where they have a transmembrane domain, or part of the protein that goes through the membrane, and then they have this receptor binding domain that's on the extracellular side of the cell. They also tend to be transporters, so as I just talked about a little bit earlier, in order for a membrane protein to be a transporter, it has to go through the membrane, therefore it has to be an integral membrane protein. One of the main examples of a peripheral membrane protein is an enzyme. So sometimes an enzyme can kind of be associated with the membrane and catalyze reactions that are either inside or outside of the cell. Because remember, a peripheral protein can either be associated with the external side of the plasma membrane or the internal side. So it's a brief bit on membrane proteins. I would really just understand the difference between an integral membrane protein, a peripheral membrane protein, and then a lipid-anchored membrane protein. All right, the last 2 things I want to talk about in this podcast are membrane dynamics and membrane potential. So as I've made clear, hopefully I've made clear, the plasma membrane is dynamic. It's always changing, budding off, fusing, growing, shrinking. In this next segment, I want to talk about membrane dynamics and specifically some of the trafficking and this budding off and fusing processes that occur in the plasma membrane. Specifically, I'm going to talk about 3 different forms of endocytosis, which is going to be phagocytosis, pinocytosis, and a type of pinocytosis called receptor-mediated endocytosis. And then I'll talk about the endomembrane system. So endocytosis is the process by which cells bring different substances into the cell. And there's a few different types of endocytosis, the first being phagocytosis. So phagocytosis is the uptake of large particles by the cell. And by large particles, I'm talking about things like bacteria, cell debris, or even intact whole cells. In contrast to phagocytosis, pineocytosis is the uptake of small particles that are in the extracellular fluid. And receptor-mediated endocytosis is just a well-characterized form of pineocytosis. So if I could summarize that in a few words, phagocytosis, big things, pineocytosis, small things, receptor-mediated endocytosis, example of pineocytosis. Let's break each of these down in a little bit more detail. So in phagocytosis, the process goes as follows. So first, the cell is going to bind whatever big object it is going to bind, whether that's bacteria, cell debris, or another cell. And this binding causes the extension of what's known as pseudopodia. And essentially, all a pseudopodia is a temporary projection of the eukaryotic cell membrane. Kind of looks like 2 little arms. And what I like to imagine is, say that this cell is engulfing a bacteria through phagocytosis. These pseudopodia are like arms that reach out and start to envelop this bacteria. In the second step, the membranes of the pseudopodia fuse to form a large intracellular vesicle. And so at this point, I like to imagine these pseudopodia basically forming like a full hug around this bacteria, and they both kind of touch, and at the point at which these arm-like projections of the membrane touch, they fuse, and now you have this large intracellular vesicle called a phagosome. Now in step 3, this phagosome moves inside the cell and fuses with a lysosome. And together, these form the aptly named phagolysosome. And what this does is this exposes the bacteria that has been taken up via phagocytosis to the enzymes in the low pH within the lysosome, which can then break that bacteria down and kill it. And this process is mostly performed by innate immune cells like macrophages and neutrophils. And one thing I find really interesting is that this phagocytosis is not just restricted to foreign material like bacteria, like I just talked about. I was reading the Cell and Molecular Approach textbook, and I came across this interesting example where it said, quote, macrophages of the human spleen and liver are responsible for the disposal of more than 1,011 aged blood cells on a daily basis, end quote. Wild. Now let's talk about this example of peniocytosis, which again is the uptake of small particles or macromolecules. That's called receptor-mediated endocytosis. And this is a pretty interesting process that goes a little bit like this. So there are receptors that bind very specific macromolecules that are on the surface of these cells, and it's typically macromolecules that you want to bring into this cell. So one of those macromolecules is an LDL particle, which is a low-density lipoprotein particle that contains cholesterol, phospholipids, triglycerides, protein, but really what the cell wants from this particle is cholesterol. So what it's going to do is it's first going to bind LDL on the surface of the cell. And there's very specific receptors that are going to bind LDL. And these receptors are concentrated in what's known as a calthrin-coated pit. And these calthrin-coated pits are essentially, they look like craters in a moon. Like if you were to look at the cell and kind of zoom in and say the cell is the moon, these calthrin-coated pits kind of look like craters in the moon, right? They're these little indents, and they're coated in this protein where this protein is calthrin. And calthrin is just a protein that coats the cytoplasmic surface of cell membranes and drives vesicle budding. And I believe it's actually clathrin, not calthrin, but you know what I mean. And I will say that about 1% to 2% of the entire plasma membrane are these clathrin-coated pits. So just keep that in mind. It does make up a significant portion of the plasma membrane. And so again, let's go back to the LDL. So we have these clathrin pits, which are just these proteins that help vesicles bud. And in that pit, there are these LDL receptors. So what's going to happen is LDL is going to diffuse into that pit, is going to bind that receptor, and this is going to cause this pit to actually bud off into the cell. And this is a process, again, that's driven by this clathrin protein. Now, at this point, what you have is a vesicle that is coated in clathrin that contains an LDL particle inside of it. And so shortly after this vesicle is created, it loses all the clathrin protein and then fuses with an early endosome. And that's going to be near the periphery of the cell. And at this point, the LDL is going to disassociate from its receptor, and now it's just free-floating LDL. Now from there, this LDL is going to move from the early endosome to a late endosome, and the late endosome eventually matures into a lysosome. And of course, inside the lysosome, you got a low pH, you got enzymes that's going to break down this LDL particle, and it's going to release the cholesterol into the cell so that that cell can use the cholesterol for a lot of important processes. So the takeaway here is that phagocytosis is the uptake of large things by the cell, so bacteria, cells, cell debris, whereas peniocytosis and receptor-mediated endocytosis is the uptake of small macromolecules or solutes. And also that calthrin is actually calthrin, which is different than Catherine, who's my friend. But really, clathrin is a protein that drives vesicle formation. It drives this clathrin-coated pit to then bud off into the cell. Let's next talk about the endomembrane system. So the endomembrane system is a group of membranes and organelles that work together to modify, package, and transport lipids and proteins in eukaryotic cells. And this consists of a few different components inside the cell. It consists of the nuclear envelope, the rough and smooth ER, it consists of vesicles, the Golgi apparatus, and lastly, the plasma membrane. And here's how all those pieces work together. So ribosomes on the rough ER synthesize and modify proteins, and the smooth ER synthesizes carbohydrates, lipids, and steroid hormones. So those are kind of like the production factories. That's where you're producing these macromolecules. And then these molecules are released from their respective ER in vesicles. Now, these vesicles fuse with the Golgi apparatus's cis face, and from there they have entered the Golgi. And at this point, these molecules travel through the Golgi. So if you've ever seen this Golgi apparatus, it kind of looks like a stack of pancakes, where each of the pancakes in a stack is called a cisternae. And as these molecules pass through the Golgi apparatus, it's passing from the cis phase to the trans phase, and it's essentially like moving through the stack of pancakes as if you were to take a knife and stab a stack of pancakes, if that makes sense. And so while these proteins and lipids and carbohydrates travel through this Golgi, they're further modified. For instance, carbohydrate groups can be added to proteins in order to tag that protein with a specific destination. So let's say that, you produced a transmembrane protein in the rough ER. Well, then in the Golgi, you could tag that protein and say, you're a transmembrane protein. I want you to go to the plasma membrane. And so that occurs in the Golgi. And once it makes it through the Golgi, it's going to bud out of the trans phase of the Golgi in a vesicle, of course. And from there, these molecules are going to be delivered to wherever they need to go in the cell. Let's say again, we made a transmembrane protein. So at this point, this vesicle that just budded out of the Golgi is going to head towards the plasma membrane, where it's going to fuse with that plasma membrane. And voila, you have a new transmembrane protein in the cell's plasma membrane. I will mention too that lysosomes are created through this endomembrane system. So you can imagine, the rough ER producing all the enzymes necessary for the lysosome and, that being packaged up in the Golgi apparatus and then being labeled as a lysosome, and then that budding from the Golgi, and then bang, you have a lysosome. So overall, the transmembrane system involves the smooth and rough ERs, the nuclear membrane, vesicles, Golgi, and the plasma membrane. And it's a system that produces molecules, mostly proteins and lipids, and then transports them to their desired location. All right, so the very last thing I want to talk about in this podcast is membrane potential. And I just want to go into this a little bit because it is important for the MCAT. So membrane potential is the difference in electric potential between the inside and outside of the cell. And at this point, you might be thinking, okay, that's great, but what is electric potential? And I talked about this a bit in the electromagnetism podcast. So if you want a deeper dive, go there. But in short, electric potential is the amount of work needed to move a unit of positive charge from a reference point to a specific point inside an electric field without producing acceleration, which you're probably thinking that's bullshit. That's super abstract. Like, how's that going to help me? Which you're right. So I like to think about it this way. You can kind of think about membrane potential as voltage, because that's what voltage is, right? Voltage is just a difference in electric potential between 2 points. And when you think of voltage, you might think of a battery, for example. And what does a battery do? Well, it drives current. It delivers a difference in voltage that can drive a current. For instance, 1 volt will drive 1 coulomb of charge carriers, which is 6.24 times 10 to the 18th charge carriers, like electrons, through a resistance of 1 ohm in 1 second. And so in a way, you can kind of think about membrane potential being similar to like a battery. If I were to stick a wire inside of the cell where, one end of the wire was inside of the cell, the other wire was outside of the cell, you would actually get a flow of electrons based upon that voltage, right? Based upon that membrane potential, you would get a flow of electrons through that wire. You'd get a current. And what does that do to? That's due to the difference in net charge inside the cell relative to outside of the cell. So the inside of the cell is typically more negative than the outside of the cell. And this is illustrated in the fact that the resting membrane potential of a neuron is about negative 70 millivolts. In other words, the voltage inside of the cell is about negative 70 millivolts less than outside the cell. And so you might be thinking, okay, that's great, right? There is a difference in charge, in net charge between the inside and outside of the cell, and that's what causes there to be this membrane potential, and if I was to plug in a wire to this cell, I could potentially flow a current based upon this membrane potential. But what role does a membrane have in determining this membrane potential? Well, technically it has like everything to do with it, right? Because the fact that the plasma membrane is impermeable to ions allows the cell to actually have a membrane potential. Like imagine for a second that you made the plasma membrane permeable to all ions. Then the second you do that, you're going to have an evening of the concentration gradients between all these different ions, and the membrane potential is going to dissipate because you're going to lose that difference in net charges, right? As all these different molecules move down their electrochemical gradients, the inside of the cell is going to look exactly like the outside of the cell. There's going to be no differences, and therefore, you're going to have no membrane potential. And so as you know, when action potentials are created in neurons, what happens is you actually open up voltage-gated ion channels and you allow these different ions to flow into the cell freely, and that's going to flip the membrane potential around. So, that just gives you an idea of what happens when you let the membrane be more permeable to a specific ion. So next time you think about resting membrane potential and membrane potential, think about the plasma membrane and its role in that. It plays a pretty important role. So that is it for the Membranes podcast. It was a really information-heavy podcast, so I apologize if some of it's kind of boring, but it is important material, and I think it is pretty high yield on the MCAT, so I hope you found it helpful. Thanks for listening to MCAT Basics, brought to you by Med School Coach. As always, if you like what we do, leave us a review on iTunes. It helps us reach more people, as well as tell your friends. Somebody is studying for the MCAT. Say, hey, I know this great podcast, especially if they're commuting or working in the lab, whatever it may be, where they can have something in their ears that they're listening to and continue studying for the MCAT. That's the goal.
Thanks for listening to Prospective Doctor's MCAT podcast. If you're a pre-med, you'll want to check out prospectivedoctor.com for tons of free tools, articles, and more podcasts that cover your pre-med life. And if you need help on the MCAT or getting into med school, check